University of Stirling, Institute of Aquaculture, for agreeing to do this webinar for us today. It's going to be on improving sustainable aquaculture through nutrition. And Monica is currently a lecturer in nutrition at the Institute of Aquaculture. She's a qualified veterinarian. She has considerable experience in fish nutrition and metabolism, particularly lipids, and the development of sustainable feeds. She has also worked on oxidative stress, general fish health, and nutritional pathology, as well as molecular biology. So I'd like to just hand over to Monica now, and thanks very much.
Thank you, Rowena, for that nice presentation. I'm gonna try and share my screen now. Okay, here we go. Sorry. Can you see it now?
Yeah, we can see.
Yes, we can see. Yeah.
Okay. So before I start, I would like to thank WiSA for giving me the chance to give this presentation today. It's gonna be, I have to say, quite light touch. I'm not gonna go into a lot of detail about results that we have got, or it's just a general idea of how I think nutrition can help aquaculture to become more sustainable. But before I start, I would like to just to give a brief background about myself, where I come from, and how I ended up as a lecturer at the University of Stirling. Well, I'm originally from the Canary Islands, so I mean, you can imagine for me it's a shock living in Scotland, but I got used to it. It's been it's been over 8 years I've been here, so now it's okay. But I'm originally from Gran Canaria. That's where I did my degree in veterinary medicine. Then after I finished that, I moved for 2 years to Sicily, to the University of Messina. It was thanks to, now it's called Erasmus Plus, I think, this program. So I was there for 2 years working on development of vaccines for a local species that was being evaluated to diversify European aquaculture, Mediterranean aquaculture. After that, I came back to the Canary Islands, back home, where I started my PhD in fish nutrition. It was about nutritional pathology. And during that time, that's something very good that they do in Spain. They encouraged you to go abroad and to go to stays in international centers. So I had the chance to go to Madeira. Also, I went to Italy, to the University of Insubria. And finally, I came to the University of Stirling, and that's how I started this collaboration with them, and they knew me, and then I was lucky enough to get a postdoctoral research assistant position here, and I've been here as a PDRA until 2017. Then briefly, I got promoted to research fellow, and then after that, I was lucky enough that there was a position opening as one of the lecturers here was leaving, and I could get this lecturer position where I am right now. So this picture here is just showing the nutrition research group. There's been some restructuring lately within the Institute, but I still consider this to be the nutrition research group. So as you can see, it's quite international. We come from many places of the world. Indeed, the Scottish are outnumbered. There is only one Scottish, which is one of the academics, which is Professor Tucker. Then we have Professor Glencross, who is the head of the group. And then the other academic would be myself. And then here on top, these are our postdocs. And then these are our PhD students here. And now we are having a new student starting next month who is coming from Egypt, Abdallah. And this slide is just to summarize a little bit my research areas. So my main research area is fish nutrition. Within that, I work quite a lot with early life stages and also with development of feeding practices for new species, for instance, meager or Atlantic bluefin tuna. But lately I've been working a lot on sustainable feeds. Then obviously there is a certain overlapping, or I like to see it like that, between fish nutrition and fish health. So how these new nutrients, how it can impact on fish health. And then lately, all this, how can impact ultimately on food quality. So for instance, how do sustainable feeds can impact on the human consumers, on what we are eating. But as I said before, I'm just gonna talk today about my vision of how nutrition can help to make aquaculture more sustainable. So the reason why aquaculture needs to be more sustainable is because we know that the world population is growing, and this increase in growth population must be accompanied by an increase in food production. Indeed, animal protein should increase approximately 50%, should double by 2050. And we know that marine-based protein can really fulfill this gap, this increased demand. The reason why is because aquaculture is a very it's a very efficient practice. In this graph, we see the amounts of feed that we need to produce 1 kilo of fish, so the FCR, and we see it's as low as 1.1 compared, so less than 6 times compared to cow, for example. However, if we need to increase the production in aquaculture, we must make sure that we are increasing the use of sustainable feeds. So what does sustainable mean? Originally, or the most basic definition of sustainability implies the use of resources at rates that do not exceed the capacity of Earth to replace them. So originally in aquaculture, we considered that the main sustainability issue was to substitute marine ingredients by terrestrial ingredients, or substitute marine ingredients. And we've seen that the industry has been doing that, okay? So if we have at this graph, this could be change aqua feeds in Norway, but it's the same trend, for instance, in Scotland as well. What we see is that the inclusion of marine ingredients, which could be the blue bars, have been decreasing steadily. So in the 90s, we would have approximately 90% of these ingredients in feed composition. However, in 2016, we have approximately 25%. So that means that we have been working on that, and we have been replacing these ingredients. Okay. However, the meaning or the definition of sustainability goes further. And for instance, it has to promote or ensure environmental sustainability. So it has in terms of climate change, biodiversity, water, and soil quality. Okay, and here in this graph, we have an example of the carbon footprint of vegetable meals that are commonly used in aquaculture, such as soybean meal. We see it's quite high. And this could be the carbon footprint of alternative protein ingredients. It could be processed animal byproducts, okay? And we see how small it is. So yeah, we are using vegetable meals. It's better than using fish meal, but still, it's not perfect. Okay, the carbon footprint is massive. We are bringing these ingredients from the other side of the world, and this is not sustainable, or we are not moving towards the zero carbon footprint, okay? And also on top of that, remember that we are contributing to deforestation. Despite of all this, still from a carbon footprint point of view, we see how farm fish are still more sustainable compared to the other livestock, okay? So how can nutrition make aquaculture more sustainable? So what we are gonna talk today is about 3 main points. So one of them, we can make it more sustainable by using protein sources that are alternative to fish and ideally to terrestrial plant meals. Also using lipid sources that are alternative to fish oil and vegetable oils, and also can boost or help to maintain the levels of omega-3, particularly long-chain polyunsaturated fatty acids. And lately, we can try to make the fish better at using nutrients, okay? And I'll briefly touch on this. We can do this through nutritional programming. So about protein sources. So here, I'm gonna mention mainly these 3. So the main alternative protein sources, apart from fish meal and apart from vegetable meal, could be animal byproducts, could be insect meal, and would also be microbial sources or single-cell protein. So here I have summarized the positive and the negative aspects that many of these sources have, but we will discuss them individually in the presentation. So for example, talking about terrestrial animal byproducts, we have a wide range of them. We have plant meal, bone meal, feather meal. They contribute in theory towards zero waste because initially these sources could be just landfill, they wouldn't be used. And there is a massive amount of these that are produced annually, over 12 million tons in South America and the USA, the main producers. However, on the negative side, we know that not all of them can be used in aquaculture. We'll discuss about this in the following slide. There is also quite a lot of limitations from a legislation point of view. And the main thing is that there is a very poor public perception, particularly in Europe. And also we might have some issues with some of them, particularly those that contain high levels of mineral or ash, such as feather and bone meal, because they can impact on digestibility as well. So there is a maximum inclusion level that we can use for these ingredients. So regarding the animal byproduct sources that can be used in Europe, basically we cannot use any ruminant source, but we can use poultry and we can use pig. But on top of that, these byproducts are classified into 3 categories. Categories 1 and 2 are considered high risk, so these cannot be used. But category 3 is low risk and can be used. The issue here is that category 3 products are fit for humans. So it means that there might be some competition with human consumption, although we humans don't go mad about consuming organs and stuff, but still, you know, that they can be used for human consumption, and also they could be used for other markets, such as, for instance, pet food. Okay, so there is, that's the downside. It's only category 3 that can be used, okay? So what happens, I was mentioning this before, there are consumer and retailer barriers that stop the use of these processed animal proteins in aquaculture, particularly within Europe. Because for instance, we know that in Chile or in Australia, vegetable meals are not as common. It's more common to have poultry byproducts, for example, in salmon feeds, that might be to have a rapeseed meal. Okay, but in Europe, we are really against this. However, and this is due mainly to the problem with the cows that we had some years ago. So that's why we are so against about feeding one animal another animal. However, recently we did a study about using poultry byproducts in salmon. It was just a survey. And what we see is that feed companies, they seem to be happy to be selling feeds that contain these processed animal byproducts. The UK consumers, they did not seem to be so reluctant and not consuming these products, so salmon fed with animal byproducts, as long as there was an impact in price. So, you know, if you give me a cheap salmon, I don't care if it has been fed with animal byproducts. However, the UK retailers were not really enthusiastic about placing on their shelves salmons that were fed this type of product. And in the end, the UK market, it's a little atypical, for instance, compared to the Norwegian one, where maybe farmers or the big, large farming companies decide what they want. So decide if I'm gonna sell a product that is high in pigments, that is high in omega-3 to this level of pigmentation. In the UK, it's the retailers. So the retailers are the ones who drive the market. So if they say, no, I don't want to sell salmon that has been fed on animal byproduct, then the farmers will stop and this won't go on. And then another thing that I wanted to notice here is that despite of this, in many UK retailers, you can find Chilean salmon. And as I said before, in Chile, it's quite common to be utilizing animal byproducts. And indeed, sometimes they are not even category 3, they are category 2. So it means that you still can buy salmon in UK supermarkets that has been fed with animal byproducts. So another option that we have here, alternative, is insect meal. Insects are quite advantageous in some ways because they have short life cycles. They can be grown on several substrates. However, and we'll discuss about this a little bit later, or well, we can discuss now anyway. It's that because right now, because of a new regulation from 2017, European regulation, I have to say, insects are considered farm animals, and because they are farm animals, they can only be fed on feed-grade substrates. So what it means is like all this of selling the insect as for bioremediation, or that they can be fed on substrate that are going to be to landfill anyway, it cannot be done because it needs to be fed on feed-grade substrate. So it means that there is competition with other livestock markets. Another issue that we have is scalability. Yeah, they have very short life cycles, but still, you know, we don't produce enough to meet the demands of the industry. And also depending of which life stage and which species, they can have very high chitin content, and chitin can impact on digestibility again. There are 7 species that have been approved to be included as a feed ingredient. but the ones that have been mainly used or monitored for aquaculture use is black soldier fly. The 3 that are here in the text box on the right would be black soldier fly, common house fly, and also the yellow mealworm. So we said that the public perception in general for animal byproducts was quite bad, but how is it in the case of insect meal? In the case of insect meal, the perception is quite positive because the consumer thinks that this is like the natural alternative because, yeah, you know, and some, for instance, this comes from the Auchan supermarkets in France. So they have a trout eating a dragonfly. It looks super nice. And what they think is like, this is what they eat in the nature. So we should go for this, okay? Also, as I said, it's sold as it more beneficial because it goes towards zero waste. Yeah, potentially it can, but right now we know that it cannot because the legislation doesn't allow it. So what it means is that the insects are competing with other livestock to get this substrate where they can be grown, okay? So an alternative would be to explore new substrates such as former foodstuff or meat or fish from categories 1 and 2, okay? And then regarding the insect farming, the good thing again is that it's not only focused for obtaining as a source of as a feedstuff, but it can also be, you can farm insects as a bioremediation, so to manage waste that cannot be treated otherwise, and also to produce biofertilizer, so the frass, the feces of the insects, they can be used as a biofertilizer. So I just wanted to mention, this is one project that finished last year, I think, Innovate UK. Sorry, okay. Sorry, someone had
Sorry, could everybody mute themselves, please? Can you please mute yourself? Thank you.
Okay. So yes, I was saying we had this project recently, and in this project we wanted to check if insect meal could be used as a functional ingredient, if it could be an additive in feeds. So in this project we worked with a company, Atomics, and what they did was to fermentate insect meal with different bacteria. And the aim was to try to generate a probiotic, or also to see, because obviously the amino acids could be like more available to the fish. So can you get like a highest growth compared to insect meal that hasn't been fermented? And we got some quite good results. So for instance, we saw that a 5% inclusion level, you could increase protein content. And also there seems to have some regulation of the immune system. We saw that at least from a molecular point of view. But one of the main issues nowadays is scalability. We mentioned this before. It's uncertain how much is produced so far because there is a lot of secretism about the producers. And also something that we know is that the price is quite high. These figures are from 2016, but should be more or less the same. So in 2016, a kilo of fish meal was around 2-4 euros, and a kilo of black soldier fly larvae was 10 euros per kilo. So quite a difference there. However, the feed producers, they are quite happy to use this ingredient. And for instance, Scretin is already including insect meal in their feeds. And also Bioman and Cargill, they have shown a lot of interest about using this insect meal. No, because I have to say also, I haven't mentioned a lot, insects have a very good amino acid profile. It's almost complete. Depending on which insect you have, you might need to substitute supplement them with crystalline amino acids, but they seem to be quite complete. The only thing is like insects have a lot of fat, particularly saturated fat, which is not very interesting from a farming or a feed producer point of view. But this can be defatted, and this fat can potentially have some applications. For instance, there is a recent study from 2020 about baking cakes and waffles with insect meal, substituting margarine at 50%. And also they did like a survey about the satisfaction of the product, and people seem to be happy to be consuming this. So potentially this sub-product can have an added value to the production of insect meal. And this is just a project that started in October this year, so quite recently. And this project, one of the main aims is to enhance the scalability. So we were saying this is one of the main issues. So this is a very large project with many partners. Among Stirling, our role is going to be to test the feasibility of insect meal in different scenarios. So as a first feed for salmon, how stable it's in a RAS system. Also, we will test it in marine water. So there are like many trials in this particular project, but the main aim is to establish the UK as a world leading center for highly automated and low cost black soldier fly farming technology. Okay, so that's just trying to address one of the main issues with insects, that it's the scalability. And just to finish, I want to talk about a single cell protein. This is a very interesting new source. Basically, it's also called a microbial protein. It's derived from different organisms. It can be either bacteria, yeast, or microalgae. Here on the right, we have an example of two main players, FeedKind that produces Calista, and then we have this branch that they produce a product called Proton. From a nutritional point of view, it's a very interesting product because it can have really high levels of protein. It can be at as high as 80% in the case of bacteria. And the amino acid profile is quite similar to fish meal. There is some freedom here because obviously depending on the conditions or the substrate that you have into your fermenter, you can modulate and manipulate the nutritional profile without doing any GM. Okay, you can also do some GM, but we'll talk about that later. It's not ideal from a European point of view market because we are also quite against GM. However, the main issues that this product has is, in one hand, the scalability, similarly to what happens with insect, the high production cost, because this is quite a new technology, so it's still improving day by day, and also we can have some issues with digestibility. So this is another project that we also got in the same call as the last insect ones that I mentioned. I'm the PI in this one, so for me it's really exciting to participate in this project. And in this one, it's led by Deep Branch Biotechnology, the company that I just mentioned. So basically they have partnered with an electric power plant that produces CO2, and they use a single cell protein or a microbe that can convert CO2 into protein. So this is pretty interesting because basically you are just cutting off the CO2. So that's really because other sources, they require methane and other gases, but maybe, CO2, we don't know what to do with it. So this is quite interesting. And then in this particular project, we will test this product both in salmon, but also in poultry. So it's like two different species, two different markets. And that the good thing is that we also have the involvement of Sainsbury's, which is one of the major retailers in the UK. So we have the whole cycle in here. So it's quite interesting and pretty exciting, and we will be starting trials next year. So, I'm really curious to see how it goes, and I'm sure it will be a very successful project. So this summary I have put, or in this table, I have put the different sources that we have talked to about. I have also included plant meals, and it's just seen from 4 different points. So environmental sustainability, consumer acceptance, legislation constraints, and also feasibility or scalability should be. We see how if they meet this criteria or not. And what we see is that so far, none of these sources are, check all the boxes completely. So probably there are things that we can do to change this. So for instance, the legislation constraints, that's something that can be changed. And for instance, scalability, that's more research, but the thing is, like, we're getting there, we're having some very nice alternatives, and maybe the solution is the combination of more than one of these, not just using one. So now we are going to talk about different lipid sources. But what I want to say here is, like, I'm not gonna talk about vegetable oils, okay? Because what I want to talk is about lipid sources that can help to boost the omega-3 long-chain polyunsaturated fatty acid level. So this could be EPA and DHA. So what we know is that traditionally, we talk about this at the beginning of the presentation, fish feeds contain high levels of marine origin ingredients that are high in long-chain PUFA and low in short-chain PUFA. Whereas now the feeds have inverted. We have a lot of vegetable ingredients, so they are high in omega-3 short-chain PUFA, these feeds. So what happens is that we've seen that there's been a steady decrease in the levels of EPA and DHA in the Scottish salmon fillet. And what does it mean? It means that if we take into account the recommendation of ISFAL, which is an agency, there is not a set recommendation of how much EPA and DHA we should have, but ISFAL is one of the most conservative ones. There are other, for instance, the American Heart Association, they recommend even higher levels, okay? So this is not too high. They recommend that we should have 3.5 grams of EPA and DHA per week. So in 2006, if you had one portion, so 130 grams of salmon, you would be getting 3.9 grams of EPA plus DHA. However, in 2016, you would only be getting 1.9 grams, okay? So what we want ideally is to see if there is any way to boost the omega-3 long chain PUFA levels in fish without using marine oils. So the alternative that we have without taking into account vegetable oil, so these could be sources that can boost the omega-3 long chain polyunsaturated fatty acid levels, could be marine multicellular organisms. So for instance, we have copepods, but copepods we know they have a lot of wax esters, which is a type of lipid class, and fish are not very good at dealing with wax esters. You can have indeed problems with digestion and well, it's like diarrhea. It's a type of orange diarrhea that it's caused by consuming lipids from copepods. You have krill. Krill is an excellent source because it is very rich in both EPA and DHA. It also has pigments, and also the lipid class is phospholipids, which are more easily assimilated, and it's very good. Then we have microalgae, and then we also have genetically modified organisms. So in this talk, because I didn't want to do it like too long, I'm gonna, I think it's gonna be a little bit long, sorry, but I'm just gonna focus on microalgae and on genetically modified organisms, okay? So regarding microalgae, they are pretty interesting because the primary producer of EPA and DHA in the aquatic environment, it's algae, okay, microalgae. So microalgae will produce EPA and DHA, and then these fatty acids will be accumulated through the trophic chains, and they will get up to humans, okay? That would be for fisheries. Then within microalgae, we have several options. We have autotrophic or heterotrophic microalgae. So basically the autotrophic ones are the ones that grow, they need light, whereas heterotrophic, they need an organic, they can be grown in darkness, but they need organic substrate. Usually the algae that are grown for EPA and DHA are heterotrophic. There are different commercial products available. Some of them have DHA only, others have EPA and DHA. We'll see how we can find them either as a biomass or an oil. However, usually when you buy an oil, in theory, it increases the price of the product. This is not very clear, but in theory, because it's a further processing of the product, and they can be used in any life stage. However, when it's used as a biomass, there is a limitation, and it's that because you can have some issues with digestibility, so you cannot include them at very high levels into the feed. So for instance, in the case of salmonids, it's recommended that not over, depending on the product as well, but it shouldn't be over 5%. Here, there are many more. This is just some examples of different algal biomass that you can have. There are many of them. And then regarding algal oil, so far as I know, there is only one product, which is Veramaris, which is produced, it's a venture, a company between DSM and Evonik. There's been some trials and they are quite successful. Indeed, we have a project. The trial should have started in May this year, but due to COVID, it's been delayed for a year, in which we are gonna use Veramaris oil together with other alternative sources, and that is going to be quite interesting. In this table, we have the content of either EPA and DHA per total of fatty acids. I'm mentioning this because it's not per total lipids, and you need to remember that all these products here, the biomass, they might contain 20% of lipid, whereas in the case of Veramaris, obviously it's an oil, so it's approximately 100% of lipid. So what we see is that, for instance, Veramaris also, the EPA to DHA ratio is quite interesting as well. It's approximately 1 EPA to 2 DHA, whereas the other products are DHA rich mainly. The thing is like, Traditionally, DHA has been considered much more important than EPA. So it's thought that if you have DHA, you don't need EPA. This is not so clear right now because we know that EPA plays very important roles, for instance, in inflammation, in eicosanoids, in resolving, so in all the inflammatory response. So it's not clear if you don't need a little bit of EPA as well. There is also some species seems to be able to retroconvert DHA to EPA, but these processes can be quite energy consuming. It's not very efficient. So ideally you want to add both EPA and DHA in the feed. Now we're gonna talk about the genetically modified organisms. So far, there is a GM yeast, and also we have several oilseed crops. These products can be rich in either EPA and DHA or a combination of both. And so far there are 3 commercial products available. So we're gonna start with the genetically modified yeast. It's a Yarrowa lipolytica. Initially it was sold as a high EPA biomass, although it was marketed as a capsule, also extracted oil for human consumption at a point. And mainly it was quite known some years ago because Aqua Chile, they decided that, or they were producing, they have like this branded salmon that was called Berlazo salmon. And basically this salmon was sold as being more sustainable because it has a fish in, fish out ratio of 1. So what they did was to substitute marine oils, fish oil, but this oil, and, and it was sold as more sustainable. This is quite, funny looking from a European perspective, how to see that a consumer in America was happier to pay a higher price because they thought it was more sustainable, but they were not concerned about the fact that these fish were fed with a genetically modified organism. And that's something that we know in Europe, usually, people who is more concerned about the environment, it goes in hand with them being against GM. So it's quite, just seeing how different the different societies, how they react to this. However, the problem with this product is that it's EPA only, and EPA only is not as well retained as DHA. So particularly when DHA levels are low. So that's what we were saying, that we ideally want to have both of the fatty acids. And also because the product that was used for fish was the biomass, wasn't the extracted oil, it means that there was a limitation in the inclusion levels. And now moving to the genetically modified oilseed crops. Nowadays, there are 2 commercial products. I think that I know for sure Latitude has been deregulated to be included in feeds. In Chile, it's being used right now. And also we have New Terra or New Seed. And so these 2 would be commercial right now. Because these are obtained as an oil is like a safest approach from the point of view, like many times when you use GM, people is concerned about the possibility of trans genes being present and being transferred potentially to the fish or to the human consumer. However, when you are working with an oil, this risk is much lower. So that's why we say it is safer. So within the 3 alternatives, we have 2 different plants. Well, they are 3 different because they are 3 different strains, or events, GM events. So we have 2 canola plants, this Aquaterra, and then we have, so the Aquaterra would be New Terra, and then we have the Latitude that it's produced by BASF and Cargill. And then on the other hand, we have Camelina sativa. This plant has been developed by Rothenstein Research, and I've been working with them for 2 years. That's how I came to the UK. My first project was to work with this plant, but we've been working quite a lot with it. So, and we have this project that I just mentioned about testing the algae oil. We are gonna test the Camelina oil as well. So how this technology works, basically in this slide, I'm gonna make it sound very simple, but believe me, it's really complex. So in this slide, we have the pathway that it's necessary to produce from short chain fatty acids, so 16 not up to 22 6N-3, that would be DHA. So this would be the pathway, and here we have the different enzymes, okay? So basically in land plants, as a maximum, you get up to 18 carbons, so you can get as high as 18,3N-3, okay? So what we are doing, or what the agronomists are doing when creating this GM plant, is to introduce algal genes, because remember, we said that microalgae were the primary producers of long-chain polyunsaturated fatty acids in the aquatic environment. So what they do, sorry, is to select genes that desaturase and elongates, so we can desaturase and elongates from 18,3 and -3 up to DHA. Okay, so basically you have your microalgae, you identify the genes, you select them, and you introduce these genes into the plant. Okay. So here we have the fatty acid profile of fish oil, which could be the black bars. And then we have the two canola alternatives, okay? The Bass Cargill, so it would be Latitude, and this could be a New Terra, would be the light green. And it's basically comparing the levels of fatty acids. So what we see is that first, despite, remember, this was a canola, so this is a rapeseed plant. So it's the same plant, and you would think maybe has the same fatty acid profile. We see how it is completely different. Okay, so for instance, the one from Cargill, it has much higher 18.2 and -6 than 18.3, whereas the one of New Sea is the other way around. And then the Bass one also contains EPA, but almost non-DHA, whereas the one from New Seed contains much higher DHA than it contains of EPA, okay? And another thing that I would like to point out here is that if you see fish oil has almost no 18 fatty acids, so very low 18.2 and -6 and very low 18.3 and -3. So in the end, if you have a look at this, what you would think is like you have a blend of oils. You are like mixing a terrestrial vegetable oil and a fish oil. So these oils can be considered as hybrid, okay? Because they have characteristic both of terrestrial plants, but also of fish oils. So that's regarding the two. So remember, these two, we said they are already commercial, at least for sure. I know that Latitude is being included in feeds in Chile. I'm not sure about New Terra. And then we have the GM Camelina sativa. So this is the plant we've been working quite extensively in the last 8 years. So Camelina sativa is a plant. It has like very interesting qualities, we can say. So for instance, it's a plant that can be grown in soils that are quite poor, even in low water conditions. It can be grown in Europe, there is no problem. Indeed, it was grown here from the Roman times, only that it was replaced by rapeseed. It could also use marginal land. And then another very interesting things like the seeds contain really high content of fat, approximately 40%. So it means that you can get out of fat from the seeds, and it's naturally rich in 18.3 and -3. And this is important because if you remember, this was the substrate that we needed to elongate up to EPA and DHA. Then the harvesting equipment that you would need to collect the seed could be the same as is used for rapeseed, for example, and other crops. And also they've done field trial in different conditions, both in the States and also in Europe, and it works quite well. It's been quite successful. Here we have, again, comparing to fish oil. So fish oil would be the black bar. And in this case, we have two different iterations of the plant. So we have one that contains high EPA and another one that contains both EPA and DHA. And what we see is that the levels are, so again, we have that fish oil will not contain almost or traces of 18.2 and -6 and 18.3 and -3, so very little. And what we see is that the plants contain arachidonic acid, which is omega-6. In the case of the EPA-rich plant, the EPA levels are higher than those of the fish oil. And then the one with EPA and DHA, we see that it contains quite a lot of EPA, also DPA, which is the intermediate fatty acid between EPA and DHA, and also quite a lot of DHA. So it's quite interesting. And then I just have this picture here just to show that phenotypically, the plant looks exactly the same. So this could be a wild type, and this could be the plant with the high EPA, so in floration and before floration, and they are basically the same. Because another thing that when they design the transgenic plant, they have a seed promoter. So it means that the only thing that changes is the fatty acid profile in the seed. Okay, if you want to analyze any other plant, any other part of the plant, they will be exactly the same. It's only the seed fatty acid profile. And again, in the same case as we saw before, this can be considered as hybrid oils because they contain both characteristic of terrestrial plants, so C18 fatty acids, but also they contain long-chain polyunsaturated fatty acids. So here, what I'm doing is just a calculation that I did about so I have like, this would be the two canola, sorry, because I gave them a different name. And here we would have the Camelina sativa, so the one with high EPA and DHA only. And here I did just some calculations. So for example, per hectare, you can produce 1.2 tons of seeds of the first canola, or 1.6 from the GM Camelina sativa. If the plants are around, the seeds are around 38% in the case of the canola and camelina, or 35% of total fat, it means that you would get from 456 up to 762 tons of oil per hectare. And from this, how much could be EPA and DHA? So here you have the calculation, and what we see is that you can get quite a lot. Okay, so from 1 hectare, you can get 124 tons of EPA and DHA. And that's a lot, and that could really complement the market. This could boost the levels of omega-3 and long-chain polyunsaturated fatty acids to levels as they were 20 years ago, okay? However, we have some barriers to genetically modified derived products. Okay, the first barrier, and this I have to say, it's particularly relevant in Europe and in the UK. Here we are really anti-GM. So the public perception of this type of product is quite poor. So it means that, you know, it's really difficult that it's gonna get accepted. I guess it has to go through education and just to perceive, because many times there is a lot of not knowledge of what GM means. You just hear GM and you think Monsanto, evil, you know, it's like it has a very negative, and many of this is because people won't really understand what it means. The shocking thing about this is that also countries that traditionally were not against GM, they are being against GM now. So there was a paper, I forgot to include here, it's from Nature in China, and they found that nowadays in China, which is a country that traditionally didn't care a lot about GM, now they are quite against. So how the perception has been worsened in the last 15 years. And then another problem that we have also, it's it could be with regulatory issues, okay? So for instance, deregulating a GM oil in Europe takes like 2 years longer than doing the same in the USA, okay? And then many times also it's very difficult to get, so for instance, we know that in Europe, in Scotland, we cannot grow GM crops, for example, and also the market is really against included GM. But also from a regulation point of view, even if the products are approved to be grown, many times the farmers don't want it again. So the last crops that GM crops that have been approved to be grown in Europe, no one is growing them, because also it's related to the public perception, I guess. So I'm just gonna briefly talk about the third alternative that we were mentioning at the beginning, that could be nutritional programming. So in nutritional programming, basically what we do is to try to elucidate an epigenetic change in the fish. So basically the theory is that during the fish development, there are like several windows in which there is plasticity. So you can elicit a change in fish. So what we do, as we work with salmon, the larvae are precocial larvae. They are quite mature when they hatch. So basically they are quite resistant to nutritional manipulation. So what we do is at first feeding, we give the fish a stimulus diet, what we call, so for example, it can be a diet that has lots of carbohydrate if we want to stimulate that route, or in our case, what we do is to give diet that have different levels of polyunsaturated fatty acids. So in that way, we'll see an example later, but in that way, we expect to elucidate a change in the metabolism of the fish that hopefully will improve the that particular nutrient utilization in fish. Depending on the species, the stimulus can be applied at different stages. So sometimes it can be applied to the broodstock. That's quite common in marine fish species because they have very tiny, the larvae are really sensitive. So if you start to try to manipulate, the nutritional manipulation, is highly likely that the mortality is going to be massive. So what you can do instead is to manipulate the oogenesis, so the formation of the eggs, by manipulating the nutrition of the broodstock. Okay. So also it seems, so I included the long-term effect because that's the main objective of the project that I'm gonna present. So right now we have 2 projects. I'm just gonna mention one just for not making too long the presentation. 2 projects that deals with nutritional programming. This one that is funded by the VDSRC. And then we have another one that it's called Aqua Impact, and it's funded by the European Union. So in the case of NutriProg, what we want to see, we have several partners, including the University of Aberdeen and Biomar. And in this one, what we wanted to see is the long-term effect of nutritional programming. So here, it's the summary of the first trial that we did, and it's just to show how we plan, how we make it work. So as we said, we have a first diet that it's called the stimulus diet. And this diet, it's provided for a certain amount of time during first feeding. In this case, because what we want to do is to elicit the net production of EPA and DHA in the fish, what we do is to use either a controlled diet that contains high levels of long-chain PUFA, so it's considered the marine diet, and then we have the stimulus diet, which in this case is a vegetable diet with low levels of long-chain PUFA. So feed the animals for 2 weeks with that diet, and then the animals are fed for a certain period of time with a marine diet. And after that, it's the challenge phase. So what we do is to move all the groups to this vegetable diet. What we've seen is that those fish that were stimulated with a vegetable diet during first feeding, they seem to recognize this vegetable diet as the challenge, and they seem to have a better production or a net production of EPA and DHA, whereas the fish that were fed with the marine diet during the first feeding, during the stimulus, they just lose EPA and DHA because of the switch to a vegetable diet. So in this particular project, the main thing that we want to see is if this effect that we know for sure occurs. So if we challenge at this point, the fish show this response. But the question now is like, will these changes, this nutritional program will last through all the whole cycle production of salmon? So that's what we are doing in this case. We have a long-term trial. Indeed, the fish are right now, we transferred them last week to marine water. And when we are just running the second trial right now, so hopefully we should have some very interesting results soon. And then also what we want to see, that's why we have the University of Aberdeen on board, is to see if these effects that we know that nutritional programming has on metabolism, these effects also can translate or can have some impact on the immune function of fish. And that's why we're working with Sam Martin from Aberdeen. They are immunologists, just to try to answer this question as well. So to conclude, this is just the take-home message. We know that nutrition can contribute to make aquaculture more sustainable. The fish meal needs to be replaced in order to meet a sustainable development on the industry. And I have to say that the salmon industry has been quite good at doing that. Remember the graph that we saw at the beginning? But this might not be enough. So we might need to have other changes, or it might not be enough to substitute simply with vegetable meal. We know that new technologies offer potential for novel sources of omega-3, long-chain PUFA in aquaculture. And all sources are valuable and can find their own niche. We saw particularly in the case of alternative protein source, how they were quite different and they didn't check all the boxes, but still they can find their own niche. And also we saw that changes in legislation are necessary. Okay, for example, in the case of single-cell protein, in the case of insects, also in the case, of GM, and in that way we can meet the UN Sustainable Development Goals and achieve zero waste. So that's it. So thanks for your attention. Sorry for this very long talk, and I'm happy to take any questions you may have.
Thank you, Monica. I'm going to start the video. Thank you for your talk. Thank you for trying to explain so many things, a lot of knowledge inside you, and I've short of breath sometimes, I think you were. But it was great.
Sorry,
Thank
I
you
have to
very
say
much.
that I'm 8 months pregnant.
Yes, I know.
So my baby is just pushing, and that's why I'm also like out of breath. So it's not I'm not very out of shape. It's just the baby pushing my lungs, and that's why I'm standing up. But still, it's tricky for me. Sorry.
No, thank you, Monica. Thank you very much. I cannot see any question on the chat box. I'll remind you that you're being recorded, but I'm very happy if you want to put your camera or unmute yourself and do the question yourself, ask the question yourself. Or I can go it through, I can do it through the chat. Will the presentation be available later? Well, I think actually I am not the best person to ask that, and probably Monica either, but I'm sure we'll make it available. Mary can answer that. It will be.
Yeah, it will be. Yeah, we'll get that sorted. Email everybody. Yeah.
Okay. I've got one question, Monica, while people is thinking about it. It was a very complex, some of the slides I had to pay a lot of attention. But one of the things I was wondering is the legislations in for the use of plants like Camelina, sativa, or all of these genetic modified plants, is the legislation stopping aquaculture on using that in the feed companies?
Yeah, it's the funny thing is that there is a lot of effort put in Europe from a research point of view and from company point of view to develop these products, but we know that it's highly unlikely that these products will be ever used in Europe because of legislation. Okay, so the theory, it sounds a little nasty, but it's like, let's create these sources that can be used in Chile, for example, so we can relieve the market and we have more fish oil to be used in Europe. That's a little bit, because so far it's like, even if there is a little bit, if the legislation changes, it's highly unlikely that the consumer and the retailer is going to accept this product. I have to say, for example, we had with this Camelina, we had a project that ended last year, and it was to use Camelina through the whole life cycle of salmon. And basically it got a lot of interest. So the BBC came to film us, and that was just a nightmare. So as soon as that went onto the news, we have members of the Scottish Parliament trying to stop the trial. It was, like people demonstrated in front of the farm, it was just terrible. Just to show, like, and it wasn't just the public, it was just the government. They didn't want to mix, our precious Scottish salmon couldn't be mixed with GM, because GM is bad. So the thing is, like, in Europe, there is a lot of limitation, not in many other countries. And that's why, indeed in Chile, it's being used right now. But in European market, it's like,
I know that I know there's a lot of misperception on those things, but I guess it will create. I could create a debate on this now, but I guess
no, and we know that's why we also try. We participate in lots of like science fair. We've been to the Royal Highland Show. Just to explain, because I think you need to start from the kids and also talk to people. And many people would come to us and they wouldn't know, why is it bad? And they don't know. It's like, oh, my nephew told me. It's like, but you need to know. Do you know that insulin is produced by GM organisms? There are like many things that people don't know. So it's very important to educate people. And also, I think just seeing how, the amount of EPA and DHA that you can produce, from these sources is like, it's something it's not neglectable, like you should say, I have the solution here, and it's just because of legislation or poor public perception due to lack of knowledge about the topic that it's, stopping or preventing us from using them.
Thank you, Monica. We have another question. What type of epigenetic change do you expect to see? For example, methylation or hydromethylation? Following these, do you expect the changes to be stable used in breeding programs?
Yeah, that's so in this Aqua Impact project, we are checking a lot. So that's why we are not sure if it's like epigenetics or if it's a compensatory mechanism. So that's why we want to see long term. I know in Stirling they've done some methylations and they have found changes with nutritional programming, but so far in salmon, we will be doing this in Aqua Impact. And yeah, and so far, sorry, what I should have said is like we've seen changes in so basically net retention and production of EPA and DHA, and that's there. And then also we've seen changes from a molecular point of view, but we still haven't within salmon investigating the epigenetic mechanisms behind that. But that's something that we'll be doing in this project. And as I was saying, also in Aqua Impact, we are working with genomic people, and they are a little reluctant. I don't know a lot about that, but how, if this could be applied to a breeding program, or if this is a trait that can be potentially lost. So that's something that we still, it's very initial phases, within epigenetics and nutritional programming.
There's another question from Guillermo. When presenting alternative protein sources, you have highlighted issues with regard to scalability and digestibility. What about setbacks on feed attractability?
Yeah. So regarding palatability of the product, so far with insects, it seems to be okay. But as I said, it's like many times you are testing diets that do not contain a really too high amount of insect meal, because first, it seems like you're going to have a bigger issue with digestibility before than you have it with palatability. But so far, the palatability, in our experience, doesn't seem to be that bad. It also depends a little bit of the species. But as I said, we've been using levels of 10% inclusion, something like that. And then for this, a colleague of mine has been working with FeedKind on single cell protein using quite high inclusion levels. And again, the main issue, to be honest, because palatability is something that can be easily corrected as well. So for instance, he was using feeds with high levels of single cell protein, but also high levels of attractants. So basically the fish were eating a lot of feed. That wasn't the issue. The issue was digestibility. So far that's the more limiting factor. So if you use the levels that are recommended, it doesn't seem to have a setback on palatability. And if you are using higher levels, it's enough to add attractants, and that can be easier, avoided.
Okay, it's now 2. There's one more question. I think that could be just the last one. So Neda says, very interesting talk. Thank you. Is the nutritional programming project you are working on the first on one of its kind?
No, well, I have to say that we started working on nutritional programming in one very large European project that is called Camelina. So in that project, there was, for instance, my original organization. They were working on CBIN, nutritional programming, and we were working in salmon. So this project was to address several issues with alternative ingredients, and it had a work package on this. And that's how, we got really interesting results in that. Indeed, that's published. So if the person is interested, she can write me and I can, or he can write me and I can send the papers to her. So we have really interesting results there, and that's how the other 2 programs came. So the Aqua Impact, the European program, came with the same partners that we have in Arena, only that we also brought these genomic people in it, and then NutriProc, it's directly built on the results that we got in Arena. So for us, at least, that was our first experience with nutritional programming, but still is quite recent.
Okay, thank you, Monica, and thank you everyone for attending the webinar. It will be available. I'm sure if we check our web, we can find it there. Sorry. And well, I hope you enjoyed, and I hope to see you soon in the next webinar. Lost my voice. Good time to lose it. Bye, everyone.
Bye. Thank you, everyone. And sorry for the long talk. Bye.